Okay, so the material in Chapter 2 continues by talking about the reactions that create and break chemical bonds. That was our topic in our previous video. So this is building on all of the information that we've done so far in the other videos in Chapter 2. And so what we have when we're talking about chemical reactions are different atoms or molecules combining and recombining. And what they're doing is they're creating chemical bonds or breaking chemical bonds to create these products. And so chemical reactions can be written as chemical equations. And so whatever we're starting with in a reaction are the reactants. The things that react are the reactants. And they will give rise to one or more products through the course of the chemical reaction. And the arrow tends to show that a chemical reaction has taken place. And then if an enzyme is a part of the chemical reaction, an enzyme is neither created nor used. It's not actually a part of the chemical reaction. It's just a catalyst to help the reaction take place. And so enzymes are usually written above the arrow. And so that's how we denote a chemical reaction. So here's what your book shows you. The participants at the start of the reaction are called reactants, and the reaction generates one or more products. An arrow indicates the direction of the reaction from reactants to products. And so over here they're giving you a visual representation, and they're showing how two hydrogen atoms and an oxygen atom react to form water. There's a physical principle. Matter is neither created nor destroyed. So we don't create or destroy atoms, we just rearrange them. We don't convert them to a different type. None of that happens. If you start with two hydrogen atoms, you have two hydrogen atoms throughout the whole process. And then instead of showing the atoms as their structures, we can just write what's happening in the chemical equation. And so your book walks you through a couple of examples of chemical reactions, one of which is a decomposition reaction, or this could be like a breakdown reaction, because we're tending to break chemical bonds and break apart a molecule that's formed. And so the most simple of that is if you have these two things joined together, we break them apart. Now, one example of decomposition reactions that we're going to talk about before the first exam is the hydrolysis reaction, where water breaks apart. So we're going to add water to this, and one of the hydrogen atoms is going to attach to the A product, and the rest of the water molecule will attach to the B product. Okay? And that's hydrolysis. Water is going to break this thing apart. The other type of reaction that we are going to talk about is a synthesis reaction, and it builds things up. This is going to form chemical bonds. And so this is the simplest example, is combining these two things to make this molecule. Dehydration synthesis is the example that we're going to talk about. Now, if you notice, this breakdown reaction and this synthesis reaction are exactly the opposite. In fact, we could draw this arrow going the reverse way. This is a reversible reaction. And so when we're going this way, it's decomposition, and when we're going this way, it's synthesis. And that's exactly how hydrolysis and dehydration synthesis are going to work. So if this was going the opposite direction, water breaks apart. But dehydration synthesis is the opposite of that. It's where we're taking this and this part of water to form a water molecule and join these two parts together. And we're going to look at many examples of that as we go through and talk about macromolecules right before your first exam. Now, as we talk about chemical reactions, one of the biggest things that we have to talk about is energy in chemical reactions. And so molecules can store energy in their chemical bonds and then release it later in places that need energy for cellular work. Okay, and when we store that energy, that's called potential energy. And then when we actually give off that energy for the cell to do work, that's kinetic energy. So your book does not assume that you've had physics before this, which is where we tend to talk about kinetic and potential energy, and there's similarities and differences. In a physics class, there's two analogies that we use to try to help people understand the difference between kinetic and potential energy. And one of those is the idea of the spring. So a spring can be compressed, and that compressed spring getting ready to spring open or spring longer, that compressed spring has some energy invested into it. It took energy to compress that spring, and so it stores energy. It has potential energy, and then you let it go and it springs open. That's kinetic energy. It's releasing that stored energy. A rubber band is the exact same way. You pull back on a rubber band, that takes a little bit of effort. That's potential energy that you've invested in pulling on that rubber band, and then when you let it go, then it releases that energy as movement. The other analogy is like a ball on a hill. You roll a ball up a hill, that you have to put some energy into that. But you've invested that as potential energy, and now that ball sits right at the top of the hill, and it's almost ready to roll down, and then as it starts to roll, it releases that potential energy as kinetic energy, the energy of movement. And so we can do the same thing in biological systems. We can put energy into some of these molecules when we synthesize them. And that energy can be stored and then later released when the cell needs to give off energy. The molecule that does that is called ATP. It's called adenosine triphosphate. And your book talks about it here and then also talks about it at the end of the chapter. Shows more of a structure toward the end of the chapter, so I welcome you to take a look at that. But ATP contains high energy covalent bonds that store energy. And then later those bonds can be broken and that energy is released for the cell to do work. Okay, and so these are examples of these dehydration synthesis and hydrolysis reactions. So we can synthesize, we can take all these little pieces and put them together in the ATP, and we've invested some energy, we've stored some energy in that molecule, and then when that molecule gets hydrolyzed, when it gets broken apart into its components, that energy can be given off for cell work. We can name these chemical reactions that do this. An endergonic reaction is the type of reaction that causes energy to be added. Energy goes in. Endo means inside. So an endergonic reaction is one that we're putting energy into. Usually from cell work or food or from ATP. The synthesis of ATP stores energy, so it's an endergonic reaction. Now, an exergonic reaction is one where we're giving off energy. Exo means out of. So an exergonic reaction, energy is released. The hydrolysis of ATP releases energy, so it's an exergonic reaction. Now, another way that energy is involved in our discussions of chemical reactions is a separate concept. Up till now, we've been talking about the fact that we can put energy into molecules to store them as potential energy, and that's an endergonic reaction. Or we can release energy from molecules in hydrolysis. That's an exergonic reaction, and that gives off energy that can be used, so that's more of a kinetic kind of energy. Now we're going to switch gears and we're going to talk about a different aspect of energy and chemical reactions, and that's the idea of activation energy, or the energy required for a chemical reaction to take place. And so activation energy is the energy required to break any chemical bonds or get a reaction started. Catalysts lower the activation energy to facilitate the reaction. And there's a number of different types of catalysts, but in biological systems, catalysts are enzymes, which are proteins that are going to help lower this activation energy and facilitate the reaction. So here's the figure in your book. Across the bottom, they're showing time, the progress of the reaction. So as time passes. And up the side, they're showing increasing energy. So things that are up here have more energy than things that are down here. Increasing energy along that y-axis. And so we start with the reactants, and this reaction progresses, and we end up with products. Okay. What they show you in the dotted line is how much energy we need to get this reaction going. And then in the red line, they're showing you what that would be if an enzyme is there to help the reaction take place or to catalyze or facilitate the reaction. So if the energy level is up here without the enzyme and it's down here with the enzyme, we say that the enzyme has lowered the activation energy. So activation energy is the amount of energy needed to begin a reaction. Catalysts can lower that activation energy, and that enzymes are those catalysts. They reduce or lower the energy of activation without being permanently changed or used up, and so they can promote chemical reactions. They can get chemical reactions to take place. Now, elsewhere in the chapter, your book tells you a lot more about enzymes. Enzymes are proteins, and so they'll have a folded or three-dimensional structure. And part of that structure is the active site. The active site are where the substrates bind. Now, substrates is a special name for the reactants in an enzymatic reaction. Okay. Chemists over here are working on chemical reactions, and they use the word reactants. Biologists are over here working on enzymes, and they use the word substrate. But when we're talking about an enzymatic reaction, a chemical reaction that an enzyme catalyzes, those two things end up being the same. So these substrates bind to the active site of the enzyme. And once they're bound to the active site, they're held together in this enzyme substrate complex, and that allows the chemical reaction to take place. That promotes or even speeds up the chemical reaction. Now the product can detach from the enzyme. The enzyme is unchanged, still has the active site, still has the same folding, and the entire process can be repeated. That active site has a specific folding to fit the specific substrates that are going to come along for a chemical reaction. Enzymes are so specific that the enzyme that breaks down sucrose, the enzyme that breaks down maltose, and the enzyme that breaks down lactose are all different enzymes, even though these sugars are very, very similar in their structure. Sucrose is made up of a glucose and a fructose. Maltose is made up of two glucose monomers, and lactose is made up of a glucose and a galactose. When we get to talking about macromolecules, we'll look at some of those structures, but the difference in some of those structures often is just the location of an oxygen atom. So the movement of a position of one single oxygen atom is enough that these enzymes can tell these reactions apart. And sucrase is the enzyme that can break down sucrose. Maltase is the enzyme that can break down maltose. And lactase is the enzyme that can break down lactose. That's how specific the folding of the active site, the structure of the active site is. It can recognize these different substrates that vary only slightly in their structure. What's another thing that you notice about the name of those three enzymes? They all end in ASE. Sugars, we tend to end their names in OSE. As you can see, of all those sugar subunits that are listed there, right there in blue. And then enzymes tend to end their naming in ASE. So enzymes are proteins. So come back to this idea once you know a whole lot more about proteins from the macromolecules video, because everything that we learn about proteins applies to enzymes. They have to be folded properly. Their shape is stabilized by hydrogen bonds. They're going to help facilitate or catalyze chemical reactions because they lower that activation energy of the reaction and therefore can affect the speed of the chemical reaction. They usually end their naming in ASE or ZYME. And they're very specific. They catalyze the formation of a product from one or more substrates, and those substrates bind to the active site. It has to be folded properly in order for those substrates to bind. Enzymes aren't changed in the course of the reaction, and they can go on to catalyze reaction after reaction after reaction. As long as there's substrates around, that enzyme can do the catalysis of the reaction. Now, because they have to be folded properly, they're sensitive to anything that breaks hydrogen bonds, which can be changes in salt concentration, temperature, or pH. And we're going to learn a lot more about how that happens when we talk about protein folding. Now, the good thing about enzymes being proteins is they can be localized to specific areas in the cell. So in Chapter 3, when we talk about cell structure and function, we're going to talk about a lot of these enzymes and where they can function in the cell. And so there are enzymes that need to function in the nucleus that can be put into the nucleus of the cell. There are enzymes that need to function in the rough ER that can be put into the rough ER of the cell. There are enzymes that need to function in the lysosome that can be localized to the lysosome. But there's even proteins that can be embedded in cell membranes so that they can localize that chemical reaction to the surface of the cell. So we can outline a little bit of what we've covered in this particular lecture. This particular video covered chemical reactions and energy related to chemical reactions. So we first said chemical reactions. We talked about some terminology. Reactants form products. We denote this by writing out a chemical equation, which uses this arrow, and the name of an enzyme can go above the arrow in the chemical reaction. So then we talked about energy, and we talked about energy in two different ways. First, we talked about the fact that energy can be stored. So we talked about the fact that potential energy is stored energy, and kinetic energy is when that energy gets released or given off. And when we store energy into a product, that's an endergonic reaction. And then when that product gets hydrolyzed and that energy gets released, that's an exergonic reaction. Okay. But then the second way that we talked about energy, rather than just the storage and release of an energy from molecules, was the energy of activation or activation energy that's required for an chemical reaction to take place. That's where enzymes came into play, because enzymes lower that activation energy and help facilitate that chemical reaction, but they're not changed. And then we looked at some other aspects of enzymes. They're very specific. They have an active site. They have to be folded properly in order to function. Anything that disrupts hydrogen bonds is going to destabilize them and denature them. So they're sensitive to changes in salt and temperature and pH. And so a lot of the things that we're going to learn about proteins and macromolecules will come back and affect what you learn about enzymes.